Good morning, good afternoon, and good evening, everyone, and welcome to our global webinar series hosted by eLab Science Bioinnovation. We are thrilled to have you, all the participants from around the world, joining us today for this advanced session on Treg cells, secrets of immune regulation. My name is Dr. Sundar Qureshi, and I will be your host for today's webinar. Over the next hour, we'll explore how Treg cells control immune tolerance, what happens when they malfunction, and how these insights based on Nobel Prize winning discoveries are shaping research and next generation therapies. Before we jump into today's topic, let me give you a quick introduction about eLab Science. We started back in 2009, and since then, we have grown into a global life science company. So currently, we have the presence over 150 countries with multiple international certifications like ISO 9001, ISO 13485, and CE. Our products have been cited in well-known journals like Nature, Cell, and Science. And we begin with the basic bioresearch reagents and ELISA kits in 2011. And over the years, we have expanded to include the cell culture solutions, clear kits, antibodies and proteins, immunohistochemistry and Western blot, metabolism assays, food safety kits and animal disease kits, and also the flow cytometric antibodies, which are also our strategic products. More recently, we have also added advanced tools like induced differentiation media, exosome isolation kits, ELISA spot, CellaQuant, primary cell isolation kits, and immune cell isolation kits as well. So along the way, we have also been recognized with awards like CITAB Supplier to Watch and the Kotler Award for the Customer Value. All of this shows our commitment to innovation and high quality research tools, supporting the cutting edge studies, including the study of Treg cells and immune regulation, which is at the heart of today's webinar. Before beginning, just a few housekeeping notes. First, to keep the momentum, a digital participation certificate and a session recording will be sent to all registrants. Attendees also have the chance to receive a free sample of selected eLab Science products. Highlights of those products are on this slide, and the full list with the eligibility and claim instructions will be sent to your registration email. Now, it's my pleasure to introduce our today's speaker, Dr. Wang Ying, who will take us through the fascinating secrets of immune regulation pertaining to T regulatory cells. The scientists who have uncovered these mechanisms earned a 2025 Nobel Prize, and their discoveries are shaping our understanding of immune tolerance and how these insights can be applied in research and next generation therapies. Dr. Wang Ying is a biophysicist specializing in the metabolic diseases and physical intervention therapies. During her PhD at Chinese Academy of Sciences, she studied how static magnetic fields can improve organ metabolism by affecting the actin polymerization, autophagy, and immune cell behavior, helping to address the diabetes and its implications. With the strong background in cellular and microbiology and multi-level analysis from tissues to cells to transcriptomics, she brings a deep understanding of cell behavior and signaling networks. We are excited to hear her insights on T regulatory cells and immune regulation today. So, Dr. Wang, over to you.
Hello, everyone. I'm Wang Ying from eLab Science. Today, I'm excited to share with you a fascinating story from the world of immunology. Regulatory T cells, secrets of immune regulation. Focus on 2025 Nobel Prize. Our report includes 4 parts. They are overview of regulatory T cells and the FOXP3, the immunosuppression mechanism mediated by regulatory T cells, diseases and disease therapies based on regulatory T cells, and the detection tools for regulatory T cells and FOXP3. Next is the part 1 overview of regulatory T cells and the FOXP3. Let's begin with the pivotal discoveries that earned them the Nobel Prize. On October 6, 2025, the Karolinska Institute in Sweden announced that the 2025 Nobel Prize in Physiology or Medicine was awarded to Mary Branco, Freda Ramsdell, and Shimon Sakaguchi for their groundbreaking contributions to peripheral immune tolerance. The foundation was laid in 1995 when Sakaguchi identified a novel subset of suppressive T cells, now known as regulatory T cells. In 2001, Branco and Ramsdell demonstrated that mutations in the FOXP3 gene caused fatal autoimmune diseases, IPEX in humans, and the scurvy phenotype in mice, thereby establishing the genetic basis of immune dysregulation. In 2003, Sakaguchi further showed that FOXP3 is specifically expressed in regulatory T cells and is essential for their development and function. Together, these landmark discoveries defined the FOXP3 as the lineage-defining transcription factor of regulatory T cells and laid the cornerstone for the modern field of peripheral immune tolerance. Building on this foundation, how do we categorize these vital cells? Regulatory T cells arise from 3 main sources. Firstly, thymus-derived regulatory T cells naturally develop in the thymus from thymocytes that recognize self-antigens with intermediate affinity. They express FOXP3, CD25, and CTLA4, have broad antigen specificity, and are highly stable, playing a key role in preventing autoimmunity. Secondly, peripheral regulatory T cells arise in peripheral tissues, such as the gut, where naive T cells differentiate under tolerological conditions. They have more limited antigen specificity, but remain functionally stable and maintain peripheral tolerance. Thirdly, induced regulatory T cells are generated in vitro from conventional T cells using factors such as TGF-beta and IL-2. They have limited specificity and are less stable, and are mainly used for research and potential therapeutic applications. Now let's trace the life journey of a regulatory T cell. Regulatory T cells development begins in the thymus, where regulatory T cell precursors differentiate into naive regulatory T cells, depending on TCR signal strength, and start expressing FOXP3 and CD25. These naive regulatory T cells characterized by CD45RA positive, CCR7 positive expression, exter the thymus and enter circulation. In lymph nodes, TCR interacts and the naive regulatory T cells become effector regulatory T cells, a process associated with increased glycolysis and the expression of activation markers, such as CD45RO positive and CD69 positive. At this stage, some cells further differentiate into central memory regulatory T cells, which retain proliferative potential and continue to circulate. After tissue-specific migration, effector regulatory T cells can become tissue-resident regulatory T cells in peripheral organs, where they upregulate survival genes, strengthen suppressive function, and express resistance markers, such as CD69 to remain in tissues like the skin, lung, or gut. Alternatively, some active regulatory T cells differentiate into effector memory regulatory T cells, which circulate and provide rapid immune regulation upon antigen re-exposure. Understanding regulatory T cells in humans adds another layer of complexity. The inhibitory function of human regulatory T cells is related to the expression level of FOXP3. Based on FOXP3 expression levels, we can distinguish three distinct CD4 positive T cell states. No FOXP3 defines conventional T cells that lack suppressive ability and produce IL-2. Expression of FOXP3 at low levels induce a limited regulatory T cell phenotype, but not a suppressive function, which is only gained when FOXP3 is highly expressed. However, human regulatory T cells are heterogeneous. FOXP3 cannot be used alone as a marker of human regulatory T cells and can be further classified based on the expression of CD45RA or CD45RO. Human regulatory T cells include CD45RO positive, low expression of FOXP3 non-regulatory T cells, CD45RO positive, low expression of FOXP3 naive regulatory T cells, and CD45RO positive, high expression of FOXP3 effector regulatory T cells. In the part 2, let's introduce the immunosuppression mechanism mediated by regulatory T cells and explore how regulatory T cells exert inhibitory functions. To appreciate a regulatory T cell's role, we must first understand self-tolerance, the immune system's ability to avoid attacking self. This is maintained through two main checkpoints, central tolerance and peripheral tolerance. In central tolerance, T cell precursors with strong reactivity to self-peptide MHC complex presented by thymic stromal cells are eliminated through apoptosis. However, despite this deletion process, some self-reaction effector T cells can escape into the periphery and become activated when they recognize self-antigens presented by APCs. Regulatory T cells deploy five primary mechanisms to enforce suppression. First is the inhibitory receptor-associated mechanisms. Regulatory T cells mediate immunosuppression primarily through inhibitory receptor-associated pathways. They interact with dendritic cells and effector T cells to limit immune activation. Through CTLA4, regulatory T cells bind to CD80 or CD86 on dendritic cells, reducing co-stimulatory signals and restricting effector T cell activation. TIGIT engages CD155 and LAG3 binds to MHC class II, further suppressing dendritic cell maturation and antigen presentation. In addition, regulatory T cells secrete inhibitory cytokines such as IL-10 and FGFR2 to directly dampen immune responses. They also promote IDO expression in dendritic cells, which suppresses effector T cell proliferation and function. Together, these receptor-mediated, cytokine-dependent, and metabolic mechanisms enable regulatory T cells to maintain immune homeostasis and prevent increased inflammation. Secondly, regulatory T cells highly express the high-affinity IL-2 receptor, which inhibits the differentiation, proliferation, and survival of TF cells by isolating IL-2, leading to their apoptosis. Thirdly, regulatory T cells express CD39 and CD73, successively converting ATP to ADP, AMP, and adenosine, and increasing CMP in TF cells through the A2A adenosine receptor, thereby inhibiting their activation and proliferation. Next, regulatory T cells produce IL-10, IL-35, and TGF-beta. IL-10 and IL-35 can induce the exhausting of CD8 plus T cells. TGF-beta inhibits the expression of pro-inflammatory factors and cytotoxicity in TF cells. Finally, regulatory T cells transport porphyrin, coenzyme A or B, and CMP to TF cells through intermembrane junction, causing damage to them, or transport CMP to directly inhibit TF cells. Regulatory T cells truly embody a double-edged sword in medicine. Their power for separation is beneficial in some contexts, but harmful in others. We will focus on the diseases and the disease therapies based on regulatory T cells. Regulatory T cells play an important role in maintaining immune balance, and alterations in their number or function contribute significantly to disease development. In autoimmunity diseases, such as SLE, RA, T1D, MS, regulatory T cells are either reduced or functionally impaired, which prevents them from suppressing autoreactive T cells. This loss of immune regulation leads to persistent inflammation and self-tissue damage, thereby promoting disease onset and progression. In contrast, during tumor immunity, regulatory T cells are often excessively enriched in the tumor microenvironment, where they strongly suppress the anti-tumor immune responses, allowing tumors to develop resistance to immunotherapy. In organ transplantation, including acute or chronic rejection and GVHD, an efficient number of regulatory T cells failed to control autoreactive or donor T cells. Harnessing the power of regulatory T cells for therapy is a major frontier. Current immunotherapies for regulatory T cells mainly focus on how to expand the regulatory T cells. There are 5 methods for expanding regulatory T cells. Firstly, in vivo expansion of N-regulatory T cells. Boosting natural regulatory T cells inside the body using agents like low-dose IL-2, ATG, or rapamycin. Secondly, ex vivo expansion of N-regulatory T cells. Isolating a patient's Tregs, expanding them massively in labor with antigen and IL-2, and then reinfusing them back. Thirdly, generation of CAR Treg cells. Through genetic engineering, engineered TCR or CAR can be transduced into N-regulatory T cells or I-regulatory T cells to eventually generate antigen-specific regulatory T cells. Next, in vivo conversion of T-com cells into Treg cells. Conventional T cells can also be converted into peripherally derived regulatory T cells in vivo, for example, by CDK8 or 19. Finally, in vitro conversion of T-com cells into Treg cells, for example, by antigenic stimulation in the presence of TGF-beta and IL-2. The typical workflow for an adoptive regulatory T cell therapy involves sorting naive regulatory T cells from blood or thymus using a CD4 positive, CD45 RA positive, CD25 positive, CD127 negative phenotype. During expansion, these cells are proliferated using APCs, and the conventional T cells can be converted into regulatory T cells by FOXP3 induction. Phosphospicity. A CAR is introduced to redirect regulatory T cells toward disease-relevant targets. Finally, combination and modifications involve gene editing or immunosuppressants to enhance stability, with applications in autoimmunity, transplantation, and hyperinflammation. The goal is to create bespoke regulatory T cell products, personalized, stable, and potent, that can restore immune balance in a variety of diseases. Beyond the cellular therapies, nature offers its own solutions. The gut microbiome is a potent regulator of regulatory biology. One groundbreaking study shows that a mixture of 17 cross-trading strains from the human microbiome promotes regulatory T cell accumulation and protecting against colitis in mice. Here, panel A shows a significant increase in the ratio of FOXP3 positive, CD4 positive T cells in mice treated with the 17 strain mix compared to controls. Panel B presents survival curves following TMBS-induced colitis, where treated mice exhibit markedly improved survival over time. Panel C reveals reduced histology sources in treated mice, indicating diminished intestinal inflammation. Together, this data highlights the therapeutic potential of regulatory T cell inducing probiotics for managing allergic and inflammatory diseases, opening the door to developing next generation probiotics. To translate these discoveries into real-world applications, accurate detections and characterizations of regulatory T cells are paramount. Researchers rely on a multifaceted toolkit. At present, a rich variety of regulatory T cell markers have been discovered, such as transcription factor FOXP3, activation and memory factors CD4, CD25, and CD127, and the CD62L. CCR4 is related to homing and origin. CTLA4, ICOS, IL-10 can provide suppressive and effective function. Human regulatory T cells can be identified based on CD4 positive, high expression of CD25, low expression of CD127, FOXP3 positive, HELIOS positive, and IL-2. Stable regulatory T cells have demonstrated DNA at the CNS2 region of FOXP3, So they can also be recognized by genomics. Regulatory T cell function can be evaluated by measuring reduced expression of co-stimulatory molecules on APCs and the ability to reduce TF cell proliferation and IL-2 production. Antigen-specific regulatory T cells can be tracked with fluorescently labeled multimers or antigen-induced marker assays. To support your research in this exciting field, ElabScience offers a comprehensive portfolio of validated reagents for regulatory T cell analysis in both human and mouse models. For example, our flow cytometry antibodies enable clear identification of regulatory T cell population. In mouse spleen, we can identify regulatory T cells as CD45 positive, CD3 positive, CD4 positive, CD25 positive, FOXP3 positive, representing 3.59% of CD4 positive T cells. In human peripheral blood, the classic regulatory T cell phenotype is CD45 positive, CD3 positive, CD4 positive, low expression of CD127, CD25 positive. It is about 7.33% of CD4 positive T cells. In summary, regulatory T cells are the major mediators of peripheral immune tolerance, and this discovery clearly deserves the 2025 Nobel Prize. They develop in the thymus, circulate in the periphery, and exert immunosuppressive functions through inhibitory receptors, cytokines, and other mechanisms, thereby maintaining immune homeostasis and protecting body. Their complex biology presents both challenges and opportunities for treating a wide range of diseases, including autoimmunity diseases, cancer, and organ transplantation. Currently, relatively comprehensive detection methods for regulatory T cells are available. Based on existing findings, further investigation into mysteries of regulatory T cells will be of greater significance for the advancement of human health. This concludes my presentation today. Thank you very much for your attention.
Okay, thank you so much, Dr. Wang, for such a comprehensive overview of Treg biology and the mechanism underlying immune tolerance. To further support the research of our customers and the researchers in this area, I would like to introduce a selection of ElabScience flow cytometry antibodies, especially designed for precise T cell and Treg characterization. What we offer here is we offer validated markers for both human and mouse studies. For the human panels, we provide antibodies targeting CD45, CD3, CD4, CD8, and CD25. Other than that, CD127 is also available. And the star of Nobel Prize, FOXP3, obviously. So all of these are key markers for identifying the total T cells, subsets, and regulatory T cells with high specificity and fluorescence stability. For mouse models, we also offer corresponding markers, including CD45, 3, 4, 8, 8, 25, 44, 45R, and obviously the FOXP3, enabling the accurate immune profiling in preclinical and translational research. With a wide range of fluorochrome options such as ELAB Fluor, Violet 450, 780, FITC, PE, APC, and some other tandem dyes, these antibodies are optimized for multi-parameter panel design, enabling a reliable detection, minimal spillover, and reproducible results. These tools provide a robust platform for your comprehensive T cell research. When you work with T cells, having a clean functional cell makes all the difference. So eLab Science also has a set of tools that will help you get there. You can start with PBMC using the separation solution, then isolate the specific T cell subsets with easy sort kits. These are also our strategic product this year. These kits are magnetic, column-free, and deliver very high purity, up to 99%, in just 15 to 18 minutes. After isolation, you can stimulate the T cells with CD3 and CD28 beads and check what's happening inside. For example, looking at the transcription factors like FOXP3. The workflow is straightforward. You can spend less time on prep and more on the science. It is also especially useful when you are working with rare or sensitive population like Tregs, where purity and cell viability are critical for your experiments. So as a reminder, I will also now want to tell you that all the webinar attendees are eligible to request one free sample from these products. Full details about the claim process will be sent to your registration email. Now we will move on to the question and answer segment. So please type your questions in Q&A panel and we will address as many as possible. If we are unable to answer all of your questions during this time, you can send them to events@elabscience.com and our team will provide you with detailed responses. Okay, the question I just received is, what are the mechanisms by which Treg cells maintain immune tolerance in the context of autoimmune diseases? Dr. Wang already mentioned that Tregs maintain immune tolerance in autoimmune diseases through several key mechanisms. Let me just summarize it for you. They suppress the autoreactive T cells by screening the anti-inflammatory cytokines like IL-10, TGF-beta, and then they also take it as IL-35, which directly inhibit the inflammatory responses. Other than that, Treg cells are also able to consume local IL-2, interleukin 2, through their higher affinity, like CD25 receptors, then starving the effector T cells to its essential growth factor and inducing apoptosis. Another thing I think she mentioned that they modulate the antigen presenting cells by expressing CTLA4, which also removes any co-stimulatory molecules from dendritic cells and prevents them from activating the self-reactive T cells. Finally, they can directly kill the target cells via granzyme and perforin pathways, ensuring immune homeostasis. So now let me see the next question. I hope this answers your question. Let's move on to the next one. Okay, Dr. Wang, next question we received is that are there any risk of a Treg therapy making someone immunosuppressed?
Absolutely. And it's something researchers watch closely. Regulatory T cells are naturally suppressive. They calm the immune system. If you give too many or too powerful regulatory T cells, you could dampen the immune response too much. They could make a person more sensitive to infections, or sometimes even allow tumors to grow if cancer cells are there. That's why regulatory T cell therapies are carefully personalized. Doses are calculated based on the patient's condition, and their immune system is monitored regularly after infusion. It's all about finding the right lock zone, not weak, not strong.
Okay, Dr. Wang, we have the next question here. Can Tregs be used for the allergy treatment?
Yes, this is a really exciting area of research. Allergies happen when the immune system overreacts to harmless substances like pollen, dust, or food proteins. Regulatory T cells have the ability to teach the immune system to tolerance, essentially telling it that's safe, don't attack. Some studies show that increasing regulatory T cells, either through cell therapy or by stimulating them with probiotics or specific compounds, can reduce allergic reactions. It's still expansional for now, but the idea is that extent of suppressing symptoms. Regulatory T cells could retrain the immune system to be more balanced over the long term.
Okay, Dr. Wang, we have the next question here. How do regulatory T cells know where to go in the body? That's really an interesting question.
Excellent question. Regulatory T cells aren't just floating around. They have a kind of GPS system. They express homeoreceptors like CCR4, CCR7, and others, which interact with chemical signals in different tissues. For example, CCR7 guides them to lymph nodes, while CCR4 might take them to the skin or lungs. This helps regulatory T cells arrive exactly where immune regulation is needed.
Okay, we have a question here. You mentioned that Treg cells can be expanded in vitro. How long does it usually take to get enough Tregs for therapy? Okay, let me just break it down for you because Dr. Wang has explained it in a really descriptive way. So just let me explain it to you because this is a very practical question. Expanding Tregs outside the body isn't instantaneous. It usually takes about 2 to 3 weeks to expand enough cells for a therapeutic dose. During this time, the cells are fed with growth factors like IL-2 and sometimes co-stimulatory signals from the antigen representing cells, which help them multiply. So the challenge is not just increasing their number, but also keeping them stable and suppressive because Tregs can sometimes lose FOXP3 expression if they are stressed or overgrown. So it is a careful balance act between the growth as well as the maintaining their function. We have another question. Can diet or a lifestyle affect Treg numbers or function, Dr. Wang?
Yes, surprisingly so. Our environment, diet, and the lifestyle have a real impact on regulatory T cells. For example, fiber-rich diets produce short-chain fatty acids in the gut, which can enhance regulatory T cells differentiation. Central probiotics like specific clostridium strains also help regulatory T cells accumulate in the gut and modulate inflammation. Moderate exercise have been shown to support regulatory T cell function as well. On the flip side, stress, poor diet, and lack of sleep can impair regulatory T cells, reducing their ability to control inflammation. So, regulatory T cells are not just influenced by labor interventions, they are also responsive to lifestyle and environment curves.
Okay. That is interesting. You mentioned that Treg heterogeneity in humans. how do we decide which Tregs to pick for therapy? Actually, it is a great observation. Human Tregs are not all the same. Some are very stable and some are suppressive, while others can lose FOXP3 expression and even become pro-inflammatory. So for therapy, scientists usually select Tregs with high FOXP3 expression and demethylated CNS2 regions in their DNA. So these are the so-called committed Tregs that are less likely to convert into regular T cells. Sometimes additional markers like Dr. Wang also mentions CD45RA. These are used to further identify the most subtle subsets. Why it is important? Because picking the right cell is crucial. Because wrong ones could either be ineffective or worse, or sometimes they also lose their suppressive function after infusion. Let's move on to our next question. How do Tregs behave differently in acute versus chronic inflammation? Dr. Wang, would you please answer that?
Okay. This is a very important point. In acute inflammation, like after an infection, regulatory T cells act quickly to prevent an overreaction. They help stop the immune system from attacking healthy tissue once the infection is controlled. But in the chronic inflammation, such as in autoimmunity diseases, regulatory T cells can become exhausted or less effective, which allows inflammation to persist. Essentially, regulatory T cells are very dynamic. They can be protective in one context, but insufficient in another.
Okay, there is another question. Is there a difference between mouse and human Tregs that we should be aware of in research? I think that there is definitely a difference. What that difference is that mouse Tregs are often easier to track and manipulate in experiments. They are more homogeneous and respond continuously and consistently to expansion protocols. Human Tregs, on the other hand, are highly heterogeneous, with different subsets showing varying stability and suppressive ability. So markers like CD25 and FOXP3 are reliable in mice, but need to be combined with other markers in human to accurately identify functional Tregs. It is also very important that translating finding from mouse to human will definitely require careful attention to these differences. Okay, we have another question, Dr. Wang. Can Tregs be used in combination with other immune therapies?
Yes. And this is where the field is really moving toward innovation. Regulatory T cells can be combined with biologics, checkpoint inhibitors, or even CAR T therapies.
Thank you. I would like to add something here for this question. Basically, in autoimmune diseases or transplant patients, you might want to boost the Tregs to prevent any immune attack. In cancer, we also might want to temporarily reduce T cells to allow other immune therapies to work better. The combination has to be carefully designed, but when it's done right, it can create much more effective and precise treatments. Okay, we have one more question here. Can T regulatory cells increase inflammation and lead to lung tumors in mice? Yeah, exactly. Dr. Wang has mentioned this in her presentation as well, that Treg cells have the capability to increase the inflammation. And they can either lead to lung tumors or skin tumors in mice. She mentioned in the second part, I think. Okay, the time for today's webinar has come to an end, and thank you all for joining us and engaging with the session. We hope that Dr. Wang's presentation, along with the overview of ELAB Science's relevant products, provided valuable insights into organelle crosstalk and immune cell regulation. If we are unable to address all of your questions during the session, please feel free to send them at events@elabscience.com, and our team will provide you with detailed responses. And don't forget to check your registration email for details on claiming your free sample and accessing the session recording. We look forward to seeing you all in our future webinars. Have a great day and happy experimenting. Goodbye, everyone.